Hi, and welcome to part 3 of chapter 4. In this section, or in this lecture, we're just going to finish up all of the different organelles that are found in animal cells. We're going to go over plant cells, and we will also discuss some of the differences between structures in animal and plant cells. So here we go. The Golgi apparatus. The Golgi apparatus is the shipping center. Personally, it's my favorite organelle. It just does lots of different things. It has a really cool shape. I don't know. It just is interesting to me. It's sometimes referred to as the Golgi body, the Golgi complex, and sometimes we just call it the Golgi. And it's composed of, think of stacks of pita bread. So it's just like this big package of pita bread, and it has the outside membrane and then the lumen area inside. Now, they are continuous with each other, but when a protein enters into the Golgi, it enters in what's known as the cis space, or here on this picture on the left, that's the top. So the cis space is usually, if you look over here to the right, the cis space is closest to the endoplasmic reticulum, somewhere in between the rough and the smooth ER. And this is because these two, the endoplasmic reticulum oftentimes is sending proteins that are in the midst of process but are not finished yet. And those proteins will be sent by vesicle to the Golgi apparatus. Now the Golgi receives everything at the cis end. Okay, so that's the receiving end. These vesicles are going to merge, they're going to fuse in the cis space. The protein or compound that's being processed is going to be released into the lumen of this region right here. When the processing is done, a new vesicle will form on the other side and then fuse with the next in the stack and go through its process here in this lumen and then come out a new vesicle, and so on and so on and so on until we get to the very last portion right here, and these vesicles are now leaving and they're going to some part of the cell. There's 6 different places that the Golgi will send things to. So this is referred to as a shipping and receiving center. The cis end is the receiving center, and the trans face or trans end is where everything gets sent out. Now, vesicles, these little bubble-like structures, are what actually transport all of these different proteins and lipids and other molecules between organelles and also between the different folds of the Golgi. So here they're showing you the places that these vesicles can go, right? So we can go to the cell membrane, it can be secretion, it can be a lysosome. If endocytosis occurs, things are brought in. They go into a lysosome, that sort of thing. Endoplasmic reticulum, nucleus, there we go. So there are 3 primary functions for the Golgi apparatus. The first is secretion. Anything that has to be secreted out of the cell will go through the Golgi and it will be packaged. Now through that processing that occurs in the Golgi, one of the things the Golgi will do will actually tag the vesicle itself that's going to be moved around. And that tag is like putting an address on an envelope. Think of those vesicles as envelopes. And all of the little protein structures or sugars or whatnot that the Golgi places on the actual outside of the package is putting an address on it so that the cell knows where that vesicle needs to go. Those that need to go outside of the cell are known as secretion vesicles or secretory vesicles. They're going to fuse with the plasma membrane because the vesicles are composed of a phospholipid bilayer, just like our cell membrane. So the membrane of the vesicle fuses with the membrane, with the cell membrane, and then just releases whatever it is it's got in its, whatever package it's carrying will be released outside of the cell. Proteins that are secreted are usually synthesized in the endoplasmic reticulum. They'll travel to the Golgi to be shipped and then to the plasma membrane for release. The Golgi also works to process enzymes and some proteins and lipids. It will continue the glycosylation process started in the endoplasmic reticulum. Glycosylation takes place in what's known as the medial Golgi, which is kind of in the center. Another process carried out by the Golgi is proteolysis. Proteolysis is the process of breaking down proteins into smaller pieces, such as when we produce insulin, we actually store it in our cells in a form called proinsulin. And proinsulin is a really long protein molecule. It's like a big long chain of insulin molecules. And the Golgi, when insulin needs to be released, the Golgi will use specialized enzymes to cut proinsulin into the smaller segments that we know as insulin. And last but not least, the Golgi's major function is protein sorting. It is going to tag these proteins and other molecules with an address somewhere that it needs to go. There are 6 different locations that the Golgi can send things to. It can either stay in the Golgi because it's a protein that's actually being used by it. It can be transported back to the endoplasmic reticulum. The ER has to use its own proteins as well. It can take it to the lysosome in animals. We're going to learn about lysosomes in a little bit. It can take things to the central vacuole, which is a structure found only in plants. It can take things to embed them in the plasma membrane, or it can send things out for secretion. So here we are, our 6 places the Golgi exports to. Stay, endoplasmic reticulum, oh, little typo there, lysosome, central vacuole, plasma membrane, and secretion. So here are lysosomes. Lysosomes are vesicles found only in animal cells. Now they contain a very special type of enzyme, and these are a type of enzyme known as acid hydrolases. The lysosome is a really fascinating organelle because it's very simple. It looks just like a vesicle. They're very small. The cell has lots of them, and they're just kind of this round membrane ball with a bunch of acid hydrolases inside of them. Now, acid hydrolases are enzymes that perform hydrolysis reactions. If you remember from Unit 1, when we put macromolecules together, such as proteins or DNA molecules or sugar molecules, when any of our macromolecules are made, they're put together using a condensation reaction where we remove water, a hydrogen atom off of one amino acid and an OH off of the other amino acid. This is known as a condensation reaction. We're removing a water molecule. But if I want to break those proteins apart, I want to break them up again, then I'm going to use a hydrolysis reaction. I'm going to add that water molecule back so that I attach that hydrogen back to where it needs to go. I attach that OH where it needs to go. And now those 2 amino acids, they're not going to bond to each other. So I can break a peptide bond with hydrolysis. Now, the reason they're called acid hydrolases is because they will only work in an acidic environment. The pH of the environment has to get below 5. And these hydrolases will only perform when it's acidic. So if this lysosome gets damaged or broken, or something happens to the lysosome inside of the cell, and it leaks or it breaks open, the cytoplasm of the cell is a neutral pH of 7. So if these acid hydrolases get released into the cytosol is a much higher concentration than these hydrolases, and those enzymes will be neutralized. They will not be able to be activated. They will be what we call denatured, and they won't be able to function. Now that's very important because of the primary function of lysosomes. Lysosomes break things down. They break down old organelles. They look for proteins that are no longer functioning, and they will absorb or fuse these organelles or molecules inside of their lumen, releasing these acid hydrolases. And when they fuse with something to phagocytize it, or to what they call endo or autophagy, when they autophagocytize it, they will turn on proton pumps, which increases the proton concentration inside of the cell, causing the pH to drop. And when the pH gets below 5, those enzymes, those acid hydrolases, are activated, and they start breaking down whatever it is that the lysosome has merged with. Different acid hydrolases are used by these lysosomes to break down different compounds. So some break down proteins, some break down carbohydrates, others break down nucleic acids, and of course lipids. So this allows the lysosome to be multifunctional, to have lots of different functions, and it needs to have that in order to kind of keep the cell nice and tidy and clean. Now, autophagy is that process of breaking down worn out organelles, and our cells in general carry out a process called ubiquitinylation. And in ubiquitinylation, any proteins or organelles or anything that are no longer functioning properly are tagged by the cell with a protein called ubiquitin. And lysosomes recognize structures that have ubiquitin bound to them. And when they see that, that is like a tag saying, hey, this guy's done. He needs to be recycled. So in autophagy, they will break down the components of an organelle and break it down into its single molecular units so the cell can recycle those molecules and use them to build new ones. And then finally, endocytosis often occurs, and this is where the lysosomes are used to actually break down substances from outside the cell. Our lysosomes are used in phagocytosis. So in phagocytosis, our white blood cells are eating bacteria. A white blood cell will surround a bacteria and engulf it and take it in and create a vesicle inside of the cell harboring this bacteria, and the lysosome will merge with it. It becomes what's referred to as a phagolysosome. And the lysosome will destroy the bacteria so it can't cause any problems to the organ. And now I had this video for you to show you the Golgi, but it's not working in this PowerPoint, so I'll play something for you in a little bit. All right, then we have vacuoles. Vacuoles have lots of different functions. Vacuoles are found only in plant cells. We don't have these in our cells. There are forms of vacuoles. We can't say they're not in any animal cells because we consider protozoans to be animals, and we do see food vacuoles and other vacuoles found in some of those protozoans. So we don't want to say they're only found in plant cells, but we don't find them in most animal cells. The functions are varied, and it depends on the cell type and sometimes what environmental conditions the cell is in. Central vacuoles found in plant cells are used primarily for storage and support. The vacuole itself is a big storage balloon, you could say, and inside of that balloon is mostly water. And the central vacuole, when a plant is healthy and has all the water it needs, the central vacuole is nice and plump. And that vacuole will provide not only storage of water and nutrients and minerals, starch in particular, but that nice plump balloon will also give the plant cells some stability. So think about when you water your plants. If you have plants at home and you don't water your plants and they have to use up all of the water that's in their central vacuole, the vacuole starts to shrink and the plant will start to wilt. When you provide water back to the plant, the central vacuole fills up. This creates what's known as turgor pressure. So it creates pressure inside of the cell, the plant cell, and plant cells have cell walls. So it kind of bulges out against that cell wall, and the plant now stands up nice and straight. So in plants, it's used primarily for storage and support. In protozoans here, in protists, it's used to expel water. So we have a food vacuole to take in food, and we have over here, these are vacuoles that are used to pump water out in case the paramecium, too much water comes across the cell. There are phagocytic vacuoles in protists. That's what that food vacuoles are. And it is believed that we have vacuoles in white blood cells. They're pretty much the same thing as our lysosomes. Now let's talk about the plasma membrane. The cell membrane itself is the boundary between everything inside of the cell and the extracellular environment. So it's very important that it has some strength and integrity and that it's very careful about what it lets come across. It's composed of a phospholipid bilayer, and we learned in Unit 1 that a phospholipid is a glycerol molecule with a phosphate and 2 fatty acid chains. Now those 2 fatty acid chains and a phospholipid bilayer, the chains are facing each other, which causes the internal portion of the membrane to be nonpolar, and the outside, either side of the membrane, intracellular or extracellular, to be polar. That makes a phospholipid in what we call an amphipathic molecule. It is polar in one region and nonpolar in the other. The plasma membrane is selectively permeable. Now we're going to spend a whole chapter on the plasma membrane, so I'm not going to go too into detail yet, but it is responsible for transporting things inside and outside of the cell, and that selective permeability allows it to be very picky about what it lets across. We don't just have diffusion or free movement of molecules back and forth across the membrane. That selective permeability, that primary characteristic, is due to the function of a phospholipid bilayer. In cell signaling, which is chapter 9, we're going to spend some time on that. We have a big assignment on that. The plasma membrane plays a major role. It's where all of the receptors are found for our cells to recognize what's going on in the outside. These receptors can be made out of proteins, sugars, and cholesterols, oftentimes in multiple combinations. And cell signaling is used for environmental responses. It's also used so our cells can communicate with each other. We want to make sure that we have good reflexes and responses. Cells communicate with each other, and they also need to recognize each other. Both forms of cell communication and recognition and environmental responses create what's called a signaling cascade. Like I said, insulin and glucose receptors. So when glucose comes into the body, we know that we need to produce some insulin. And so the pancreas receives that glucose signal and starts pumping out insulin to assist with glucose metabolism. And finally, the plasma membrane contains lots of proteins in it for cell adhesion. Our cells stick to each other, but they do so in such a manner that they're not just jumbled up together in any old way. They have a very distinct organization. Think about it. Everybody's kidney is the same shape. Everybody's liver is the same shape. We all have the same shape of heart and brain. So that's what works properly for that organ. And so these cells must adhere to each other, to some kind of matrix, in order to create these structures. These cell adhesions are also used for cell recognition, and different types of adhesions. We're going to look at the different cell adhesions in 10.2. They are also used in sharing of nutrients and in cell signaling. So here we have our plasma membrane with a signal cascade. Here we have two cells are adhering to each other, right? The top is cell 1, the bottom is cell 2. So two proteins embedded on the membrane, on the outside of the membrane here, are sticking together. So these two cells can stick together and be attached. Then we have here a transport membrane that's bringing glucose into the cell. Right? Our cells need glucose for energy. And over here we have another protein. Now notice this protein is a signaling protein. It's a receptor. It is not carrying anything into the cell, but when some type of outside signal, when some kind of outside signal binds to it, it's going to cause it to change shape and release these proteins that interact with each other in it like a row of dominoes. And it's called a signaling cascade. And eventually one of these proteins is going to go to the nucleus and cause some kind of cellular response to occur. Now, the semi-autonomous organelles consist of the mitochondria, the chloroplasts, and the peroxisomes. We call them semi-autonomous. They can grow and divide, but they're not totally autonomous. Now, mitochondria are found in plant and animal cells. Peroxisomes are found in plant and animal cells, but chloroplasts are only found in in plant cells. Now, the mitochondria is most often referred to as the powerhouse or the power plant of the cell. It is where the majority of a cell's ATP is made. It is membrane bound, and it has two membranes, but it is not considered part of the endomembrane system. It consists of both an outer and inner membrane. It has an inner membrane space, which is just kind of the space in between the two membranes. And then inside of the most inner membrane, it's known as the mitochondrial matrix. The inner membrane is folded. It has lots and lots of folds in it, and these are referred to as cristae. And these folds are very important in order to increase the surface area for ATP production. That inner membrane is critical in ATP production. So the more inner membrane that a mitochondria has, the better able it is to produce ATP. And that is its primary function. It converts chemical energy into forms of energy that can be used by the cell. It can be used for synthesis, modification, and breakdown of cell molecules. Not a whole lot, but it can do some. It's responsible for housing the electron transport chain that we're going to learn about in the next unit. And it can generate heat in what are referred to as brown fat cells, and they are used in thermal reactions in the body. So here we have our mitochondria. You can see this inner membrane here has all of these infoldings, these cristae, just like the surface of our brain. And it serves the same purpose. We need to increase surface area. The more surface area our brain has, the smarter we are considered to be. So we have lots and lots of cristae in the brain. Well, the same thing is occurring here in the mitochondria. The more cristae we have, then the more surface area of cell membrane. Of inner membrane for housing the electron transport chain to make ATP. Chloroplasts. Chloroplasts are found only in plant cells, and they are used for photosynthesis. They are the primary structure where photosynthesis occurs in plant cells, and it is also composed of an inner and outer membrane. There's a third membrane inside the lumen, inside of the actual chloroplast itself, called thylakoids. And these thylakoids are these individual stacks. So we have these big membranes in here that's referred to as the thylakoid membrane. And the thylakoid membrane folds in on itself, more surface area, and it creates what are called a granum, a large, think of a stack of coins, looks like a stack of coins. So a granum is the whole stack. A thylakoid is each individual each individual coin that makes up the granum. Its primary function, again, photosynthesis, converting light energy into chemical energy. This is all going on inside of the chloroplast. And we'll spend more time on the chloroplast in the next unit. Here's just a close-up again of a granum that's a stack of these thylakoids. Here's another close-up picture of a chloroplast inside of the cytosol. Now that brings us to peroxisomes. Peroxisomes are very small organelles. Where they came from and why we have them, nobody really knows. Their primary function is breaking down molecules, especially lipids. They catalyze chemical reactions as well. They break down molecules by removing a hydrogen or adding an oxygen to it. And these types of reactions create a waste product known as hydrogen peroxide. Hydrogen peroxide is highly reactive inside of our cells. It is considered a free radical. So the peroxisome has to break down the hydrogen peroxide that it made, and it does so with an enzyme called catalase. And catalase breaks down hydrogen peroxide into water and oxygen gas. And if you ever poured hydrogen peroxide on a cut, the catalase in your red blood cells is what is causing majority of the fizzing that you see with hydrogen peroxide on a wound. So I'm going to just show you a quick video here. This is from med school, but I think you can get the gist of it. You get the general idea of how it works.
Peroxisomes. Peroxisomes are 0.5 micrometers in diameter and are surrounded by a membrane. The highest concentration of peroxisomes is in the liver and kidney. Proteins are directed to the peroxisomes by a unique signal sequence with the help of protein chaperones, peroxins. Functions: generation and degradation of hydrogen peroxide. It contains enzymes that can either produce H2O2 or break it down. Long-chain fatty acid metabolism, detoxification of harmful chemicals by superoxide dismutase, synthesis of plasminogens and platelet-activating factors. The marker is uric acid oxidase. Peroxisomal disorders: Zellweger syndrome. Zellweger syndrome is also known as cerebral hepatorenal syndrome and is the prototype of peroxisome biosynthesis disorders. It is characterized by craniofacial dysmorphism and profound neurologic abnormalities. Zellweger syndrome is the most common peroxisomal disorder to present in early infancy. Genetics. The peroxisome biogenesis disorders, including Zellweger syndrome, neonatal adrenoleukodystrophy, and infantile Refsum disease, are inherited in an autosomal recessive manner. They are caused by mutations in at least 12 different genes. The majority of infants with these conditions have mutations in either the PEX1 or PEX6 genes that encode ATPases needed to import protein from the cytosol into peroxisomes.
So I wanted to show you that video because I wanted you to see that here we have one of the absolute smallest organelles that has minimal function compared to most of the other organelles in the cell, yet if it doesn't function properly, it can cause some really serious problems very early on.